Good morning to all. My name is Fatima from SEM 5 and my topic is studies with microorganisms and tissue culture. So today we are going to discuss on the importance of microorganisms and study in depth about them. Studies with microorganisms. A tiny organism that can only be seen through a microscope are called as microorganisms. One such microscope is a compound microscope. Second, most microbes are around 1 micrometer in size. Third, an average lifespan of a bacterium is around 12 hours. Fourth, microorganisms have a very important role in contribution to digestion, making complex particles crushed down to simpler ones. Such example is of Lactobacillus acidophilus, which helps in degrading larger components to smaller ones. Fourth, promoting production of vitamin K, detoxifying harmful chemicals and promote development of immune system. They play a crucial role in training and development of major components of host innate and adaptive immunity system and are also essential for the nutritive growth. Last, the nutritive requirements are simple. They can be grown rapidly in the laboratory in pure culture in large quantities and this makes them easier to handle. Now let's come to recombinant DNA technology. Microorganisms are used as tool in study of drug metabolism. Recombinant DNA technology is important in production of vaccines and protein therapies such as for human insulin. Second, they are routinely transformed into drug-producing factories using a technique known as recombinant DNA technology, in which DNA from different species is combined and inserted into a host organism. One such example is insulin. Fourth, recombinant DNA technology involves direct manipulation of an organism's gene. This leads to favorable and desirable quality and quantity. By inserting our desirable sequences, this helps to produce variations. Last, this technology alters the genetic makeup of an organism. So here you can see the production of insulin in which first for humans who have deficiency of insulin in any form or diabetic disease, the blood glucose level rises quickly. Such patients are to be given insulin injection in order to maintain their blood sugar level. This insulin have to be manufactured in the laboratory using recombinant DNA technology. In this technique, we isolate the insulin gene from human. In the next step, we isolate the plasmid from bacteria. This plasmid is added with insulin gene to form a recombinant plasmid. This recombinant plasmid not only has properties of bacteria but also a precious property of insulin. Production of this recombinant plasmid is then inserted into bacteria, which is known as transgenic bacteria. In this next step, large-scale production of bacterial culture takes place due to increased number of transgenic bacteria. The quantity of insulin also gets high and insulin is also produced in high quantity. Thus, insulin can be extracted, purified and can be injected into the patients suffering from diabetes. Tissue culture. Cells and tissues can be grown in accurately defined media such as agar, which can provide them with essential nutrients for growth and development. Prolonged preservation of living cells outside the body can be achieved by a technique known as tissue culture, in which they are grown in laboratories as they require sterile, optimal conditions for growth. Tissue culture is a method of biological research in which fragments of tissues from an animal or plant are transferred to an artificial environment in which they can continue to survive and function. Fourth, a large number of different cell types of several mammalian cells, including the humans, have been grown in tissue culture. This technique helps in study of viruses and viral and chemical alterations. Now let's come to our last topic, advantages and disadvantages. First, microorganisms have been very useful in study of mechanism of different metabolic processes. Mechanism of metabolic inhibition by antibiotics can also be studied by microorganisms. Second, due to selective fermentations of different sugars by different microorganisms, yeast and bacteria have been used in the estimations of individual sugars and mixtures of sugars. So carbohydrate fermentation is a process in which microorganisms are used to produce energy. Chances of chemical and microbial contamination are very high in mammalian culture. As animal cells are highly nutrient, so they attract microorganisms and spores, which leads them to exposing themselves to certain microbial contaminants. They are high maintenance because as they require sterile conditions, optimal temperature, different plant hormones to provide them sufficient nutrients for growth. Last, perhaps the most difficulty encountered in such studies is the tendency of animal cell to differentiate during multiplication, because the culture which is derived is not identical to the cells in vivo. So that was all about studies with microorganisms and tissue culture. That concludes my topic. Thank you.